Okay, well, we're going to continue on. We were talking about, of course, after RNA processing. She's going to stab me with that? I'm getting scared here. I already got stuck today. They stuck me for 5 hours yesterday. No. Yeah, well, okay, you don't have poison in that, do you? Good. Okay. Well, it looks like one of those things they put into me. Okay, but at any rate, folks, we left off on Tuesday with this idea. Once we finish processing RNAs, now there are new sequences and there are old sequences that have been eliminated. Those sequences bind proteins or don't bind proteins. And those proteins are what the export machinery of the nucleus recognizes. It tells that export machinery, I have been fully processed. Now get me out of this place. I've served my time, okay? I need a parole. So now the nuclear export machinery will take that out. So what we have to do is take a look at how do we get things in and out of the nucleus. Because you not only have to get things out of the nucleus, like messenger RNAs, ribosomes, stuff like that, we also have to get things into the nucleus. For instance, don't forget, proteins are synthesized on ribosomes, which are located in the cytoplasm. Yet the nucleus is full of proteins. So we have to get those nuclear proteins from the cytoplasm into the nucleus. That means we've got to cross two nuclear membranes. Because the nucleus has a double membrane. An outer membrane, which is very different from the inner membrane, is continuous with the endoplasmic reticulum. And then an inner membrane. Yes?
On the inner membrane and the outer membrane, does the port that lines up from the inner membrane and the outer membrane the same hole?
Yeah. In fact, we're going to see that. I'm going to go nuclear pore structure in just a moment. So, yes, absolutely. It spans through both of them. Okay, well, what gets things in and out of nuclei? Okay, well, the nucleus is studded with these structures that are called NPCs, nuclear pore complexes. Now, a lot of people just call them nuclear pores. That's legit, but I don't particularly like the term nuclear pores because it makes you think there's just a bunch of holes in the nucleus, in the nuclear membrane. It's not. The nuclear pore complexes are very large, complex assemblies of well over 100 different proteins, forming a very big, very complex structure. In fact, the proteins that make them up are called the nucleoporins. And there are at least 100 different kinds, some say more than 200. Okay, so it's clearly a complex structure. So I'm going to make kind of like two little drawings of what these things look like. These nuclear pores are what allows things across the nuclear membranes. And as we're going to see, the nuclear pores will allow passive transport, in other words, diffusion of small molecules pretty free. Non-selectively. If you're small enough, you can go right through it like it isn't even there. But large stuff, like proteins, RNAs, and things like that, that requires specific transport. In other words, you have to recognize certain features that either are a ticket into the nucleus or a ticket to freedom from the nucleus. So, first, if we were to look at a top view of a nuclear membrane, what we would see are these structures, and they're about, I think they're about 100 or 200 nanometers. That's huge in molecular terms. About 1/10 to 2/10 of a micrometer in size. They're enormous structures. We have these 8 large balls. 4. And then some kind of central ill-defined substructure and what seems to be some kind of connections to these balls and around these balls. So that's currently what these things look like. Okay, now if we look at it in a cross-section, we see that the nuclear pore complexes span both membranes. So here's the outer and here's the inner nuclear membrane. Now we have these balls on both sides, and it appears that the nuclear membrane kind of folds in like so. Here we have these balls again. So and this is the membrane, of course, and here's the space between the two membranes. And then at least in the electron microscope, you see some kind of hazy-like material and then a sort of central large something or another. So this is more like a cross-section. Now the other thing that you see, at least with certain specimen preparation techniques, is you see these kind of coming off these balls, you see these little kind of extensions of protein, sort of like so. Those are some of the nucleopomerins, and they play roles in. And there also appears to be connections to sort of a basket-like, latticework basket-like structure, kind of like so. And this basket is then connected underneath to a protein filament network called the nuclear lamina. Okay, so they're fairly complex, large structures. Now what we see here is that these nuclear pores have two types of transport channels. There are these small, and they appear that they're probably located around sort of the outer rim. We have these small, what we could call passive transport channels. And these are nonspecific. Now they allow diffusion from things from high to low concentration through these channels. These are apparently small. Best test is they work really good for small molecules. Things like ions, metabolites, ATP, nucleotides, that kind of stuff goes in and out without any problem. When you start talking about things the size of proteins, it slows down sharply with size. And it seems like they have an absolute cutoff of about 30 to 50,000 molecular weight. And things that large go through these very, very slowly. Okay. Well, clearly a lot of things that you want to get in and out of the nucleus are an awful lot larger than a few tens of thousands of molecular weight. A messenger RNA with the associated proteins can have a molecular weight in the millions. So we're talking very big. Same thing with ribosomal large and small subunits. Those entire ribosome components are carried out as a single piece out through the nuclear pores. And these have molecular weights of about 2 or 3 million. They're huge. So we had to have something else. And that's where this central thing seems to be. This is where we have a single specific channel that recognizes certain things having essentially an import or an export sequence, a ticket in or out of the nucleus, and will allow things to cross through that. That's for the big stuff. So we got these two types of channels, the passive channels and then the much larger specific channels.
And the small channels are they that represent the dots?
Yeah, the best model of these is that there's a whole bunch of small channels, maybe one for every ball, in that kind of middle, in that kind of peripheral area. Okay, all right, so that's basic structure of these nuclear pore complexes. Now, how do we get things in and out of the nucleus? Well, we're going to, somehow this nuclear pore machinery has got to somehow recognize certain features that are effectively a ticket in or out of the nucleus. What we could call export signals, getting things out of the nucleus, and import signals or sequences that get things into the nucleus. And the way most of this works, this is not the only mechanism, but this is the predominant mechanism, is we use a small GTP binding protein. In fact, what we call a small G protein. And that G protein goes by the name of RAN. We're going to see this in just a second. Now, like I say, this is not the only mechanism, but it's the most widely used one. Okay, major mechanism uses a single subunit or monomeric G protein called RAN. Now, RAN is a distant relative of that G protein RAS that we find in a very important signaling pathway. In fact, we're going to cover that. Those of you who took cell with me, you remember us going through the RAS MAPK pathway. We'll go through that again because of its importance in development and cancer biology and all kinds of fun things like that. But anyway, RAN is a distant relative, a small single subunit G protein. And like all G proteins bind either GTP or GDP. They have one structure when they're bound to GTP and a different structure when they're bound to GDP. And as a matter of fact, let's say, oops, I'm going to keep my usual convention here. So here's a G protein bound to GTP. Here's the same G protein bound to GDP. You notice it exists in two different, distinctly different structures. I'm trying to keep the same convention just so it doesn't cause confusion. Okay, so with GDP, it's this structure. It switches its tertiary structure when it has GTP bound to it. And we can go back and forth. We can do an exchange of GDP for GTP. Now this is an exchange where we dump the GDP out, we expel the GDP and pick up a GTP. G proteins cannot make GTP from GDP. They're just not capable of doing it. Does anybody know why? How do you make GTP from GDP? You have to covalently attach a third phosphate group to it. Problem is, what kind of reaction is that when you attach a phosphate to something? Phosphorylation, and it's a condensation reaction. It's extremely energonic. It takes a lot of energy to do that. So this guy cannot do that, can't provide the energy to do that reaction. So it has to kick the old one out and pick up a fresh GTP. Now what it can do though is once it has GTP bound, it can hydrolyze the GTP. And of course hydrolysis is an energy releasing or exergonic reaction. So all I have to do, so these enzymes, these G proteins are weak catalysts of GTP hydrolysis. Not very good at it, but they will do it if you leave them around long enough. So if you have GTP bound, you eventually hydrolyze it into GDP, you switch back to this state. That's the common thing with these G proteins. Okay, and RAN is one member of this, and its purpose especially to get things in and out of the nucleus. Because as we're going to see in a moment, the RAN protein acts in effect as a shuttle bus. When it has GTP bound to it, it carries things in one direction, and when it has GDP bound to it, it carries things in the opposite direction. So what direction it goes, whether it goes in or out of the nucleus, depends on whether it has GTP or GDP bound to it. So that's a kind of neat little thing. All right, well, let's take a look at this kind of mechanism here. What we're going to do is I'm going to draw a nucleus in cytoplasm and a boundary between the two. Okay, so what we will do here is two membranes, and we're going to have a nuclear pore over here. There's a nuclear pore, and we'll have another nuclear pore over here. Okay, this is inside the nucleus, and this is inside the cytoplasm. Okay, now what we've got inside the nucleus is going to be RAN with GDP attached to it. Okay, now in that form, it can bind a number of proteins that are known as export receptor proteins. And these export receptor proteins act like adapters. On one end, they bind RAN with GDP. They cannot bind RAN that has GTP on it. Only RAN with GDP. They only recognize the circular shape. So we'll call this an export receptor. Okay, now it's a protein. The export receptor is a protein, or it's a complex of proteins. Now, on the other end of these export receptors, they bind and recognize certain sequences on proteins and possibly certain nucleic acid sequences. A little debate about that. But these sequences, we could say, are export sequences, tickets out of the nucleus. So you can picture here, this will recognize things that have the appropriate tickets out of the nucleus, the appropriate export sequences. That can include things like fully processed messenger RNAs. It has proteins attached to has an intact cap, and has proteins attached to poly A tail, all this kind of fun stuff. So, we're going to bind to some kind of cargo. I'm going to, whatever you call it. We'll just call it a cargo. Okay. We have assembled this export complex. Ran with GDP, an export receptor that acts as an adapter. Question?
The book says GTP.
I'm sure. Oh, you're right. I'm sorry, you're absolutely right. Oh boy. My vision. Okay, sorry, this is ran with GTP. Okay, I'll put it like that. Okay, I'm sorry about that. It's just bad. I looked at that and bad eyesight. Okay. All right. Yeah, that was my mistake. Okay, that is my mistake there. All right. Ran with GTP. GTP takes you out. Let's put this one down. Boy. All right. Okay, sorry about that mistake there. So we switched all that to ran with GTP. All right. Thanks for pointing that one out. That's what happens when you get old and your vision starts going. All right. Well, now we've got this export assembly ran with GTP, the export receptor, and whatever kind of cargo we've got. Now what's going to happen is certain proteins, and it seems that those extensions actually bind this in some way. It's been photographed in the electron microscope. Remember we said it's kind of like these protein extensions coming out of these balls. That actually seems to be able to somehow recognize and bind these export receptors in these whole assemblies. So we bind it here, and our export receptor, and the cargo, and then somehow it gets it through that main channel, the whole assembly, and out into the cytoplasm. So now we're going to be out in the cytoplasm. Okay, we just carry things out of the cytoplasm. Now recall, ran with GTP. It's a G protein. It's a G proteins hydrolyze their GTP. And not only that, there are proteins in the cytoplasm that are called GTPase activating proteins or GAPs that can speed that hydrolysis up. So let's take this little regulatory protein, we'll call it GAP. This little protein here binds to the ran with GTP. It stimulates that G protein to hydrolyze its GTP real fast. It'll hydrolyze it on its own if you leave it around long enough, but this speeds the process way, way up. So now what happens is ran does GTP hydrolysis. And now it goes to the GDP state. Now the problem is, ran with GDP has a different structure than ran with GTP. So now at this point it can no longer hold on to the export receptor. So the export receptor is released. And not only that, when the export receptor releases from the ran, it will also start to release the cargo. So whatever we took along, our messenger RNA or whatever, is now released. So we've taken that whole export complex. Once it gets into the cytoplasm, the GTP is hydrolyzed, and now that whole assembly falls apart. Question?
does that say, RAN? Is that RAN?
Yeah.
Okay,
Uh-huh.
I didn't know if you changed it to RAS.
No. It's related to RAS, but it's not RAS. It's a close relative. Okay, now we've got our RAN with GDP on it. It turns out that this can bind a second set of proteins called import receptors. And the import receptors will recognize and bind molecules that have nuclear import sequences. Now, we'll see what those things are in a minute. So let's put an import receptor here. And it's attached to some kind of cargo that has an import sequence. Now we have a little bit of idea about these import sequences. In proteins at least, they tend to be short, few tens of amino acids long in a protein, and full of hydrophobic amino acids. But there's no one single import sequence. They have features. Most of them have one hydrophobic stretch of amino acids, a short space, and a second hydrophobic stretch. But there's no specific sequence that all has. They have certain features, and those hydrophobic stretches exposed on the surface of the protein seem to be part of that. So it seems to be common elements of import sequences. Now you can regulate a protein and say, ordinarily I'm in the cytoplasm, but then I'll go into the nucleus by altering its shape and either burying an import sequence into the inside of the molecule where nothing can find it, or exposing it on the surface. And we already have an example of something like that. It's a class of zinc finger transcription factors called the steroid hormone receptors. Without the steroid hormone, they're in the cytoplasm. And they have a nuclear import sequence, but it's buried inside the molecule, so this import receptor can't recognize it. It's hidden. But now, along comes the steroid hormone. It binds to the steroid receptor, causes the receptor to alter its tertiary structure, and now that import sequence is exposed on the surface. And it can bind to these import receptors, and lo and behold, the steroid receptor and the steroid hormone attached to it now gets shuttled into the nucleus. And of course, that's where transcription factors belong if they're going to be regulating gene expression. So that would be one example. So now we have our RAN with GDP recognizing and binding import receptors with some kind of cargo. In fact, one is called, they've identified a couple, and one is called importin. That's one example of these import receptors. That's one of several import receptors. And here again, they act as adapter proteins. Each import receptor will bind RAN with the GDP, but not RAN-GTP. And each import receptor recognizes certain types of import sequences. Okay, so now we bind this thing, and now RAN goes the opposite way into the nucleus. So now we've carried our whole assembly into the nucleus. Here's our import receptor. I'll just put in. And here's our cargo. Okay. Our next step is now we have to break that complex apart and release the cargo and release the import receptor. And then we have to switch the RAN with GDP. We have to do that exchange of GDP for GTP. So here again, we have another protein. They're called Guanine Nucleotide Exchange Factors, or GEFs. It's a regulatory protein. We find this one in the nucleus. Now these guanine nucleotide these guanine nucleotide exchange factors, the regulatory proteins, what they do is they bind to G proteins, at least the appropriate G protein, and then they stimulate that G protein to kick the GDP out and grab a GTP. In other words, to exchange an old tired GDP for a brand new fresh GTP. Now the G proteins can do that by themselves, but they're really crappy at it. They're very slow at it. It takes them like 10, 20 minutes to do that on the average. It can be done in a fraction of a second if you have these exchange factors bound to it. So what we're going to do here is now we're going to stimulate this guy to exchange, to kick out the GDP and pick up a GTP. And when that happens, now RAN goes back to the GTP state. And lo and behold, import receptors cannot bind to RAN with GTP. Export receptors can, but import receptors can't. So now we release the import receptor. And that in turn releases the cargo into the nucleus. And of course now, the RAN with GTP can bind a new export receptor. So we got this constant shuttle buzz. GTP takes cargo, RAN GTP takes cargo out. We convert it into RAN, we hydrolyze the GTP, goes to RAN GTP, it releases the cargo, and now picks up stuff to take into the nucleus. It goes into the nucleus, and then once again we release the cargo and go back to RAN GTP. So we do this shuttling back and forth. And this is the major mechanism of nuclear and cytoplasmic transport. There are some other mechanisms, but this is the major one. That's the one we'll stick at.